Good morning. My name is Francesca Capon. Thank you very much for inviting me to give this talk. These are my financial disclosures. So we all know that the presentation of generalized pustular psoriasis is very different from that of plaque psoriasis. But with this slide, I wanted to highlight the fact that these two conditions are also very different at the genetic level. So plaque psoriasis is considered a multifactorial disorder caused by several susceptibility alleles, which are common variants of small effect size, that then interact with environmental factors such as infection, stress, or trauma. Generalized pustular psoriasis, on the other hand, behaves a lot more like a monogenic disorder. I'm calling it near monogenic now for reasons that will hopefully become clear during the course of this talk. And although there is a role for environmental triggers in GPP, we also know that in most patients the genetic component of the disease is driven by mutations in the single gene. And these mutations are rare, deleterious variants of large effect. So the first such gene, the first GPP gene to be discovered was the L36RN gene, and this happened more than 10 years ago now when my group and the group led by Hervé Bachelet found that patients with generalized pustular psoriasis carry a recessive mutation in the L36RN gene. And over the years, by screening large numbers of patient datasets, we found that these mutations are seen in roughly 25% of GPP cases. And interestingly, a small percentage of affected individuals carry single heterozygous change, so just one L36RN mutation. And we know, so L36RN encodes for the L36 receptor antagonist and labeled as L36RA here. And this protein is a negative regulator of L36 signaling, which can bind the L36 receptor and shut down signaling from the receptor. So in the absence of this negative regulator, when you have L36RN mutation, the brake is released from the system, and what you have is uncontrolled L36 signaling and excessive production of cytokines such as IL-1, IL-6, and IL-8, and these cause the cutaneous and systemic inflammations, which are the hallmark of GPP. So I said earlier that L36RN mutations are observed in roughly 25% of individuals with generalized pustular psoriasis. And something else that we've learned over the years by screening patient cohorts is that these mutations are found across all ethnic groups. In fact, we now know of several founder mutations which originated independently in Europe, North Africa, and East Asia, and which then expanded in those geographical areas and became the dominant mutation in those continents. So, for example, in European population, this S113L mutation, which you see at the bottom of the panel, is the dominant GPP allele. In North Africa, we've got this proline to leucine substitution, which was first described by Hervé Bachelet and his team, and so on and so forth. There are also rarer changes that have been observed maybe in a couple of patients in the literature, or sometimes even in a single case. But even in these cases, what is striking is that these mutations have been observed globally. So individuals with L36RN alleles have been described in East Asia, South Asia, Latin America, the Middle East, etc. The problem with this rarer allele is that when you have a nucleotide change that is seen in just a couple of patients, or in some cases even in just one, it's can be sometimes challenging to determine whether this is a benign polymorphism or a mutation. And this especially if the nucleotide change has been identified in a clinical setting which doesn't have the facilities or the expertise to functionally characterize these alleles. And of course, this characterization, this classification of a variant as a benign polymorphism or a pathogenic mutation matters for a correct diagnosis of the disease. And so to investigate this a bit further, my group has been systematically characterizing at the experimental level all the L36RN mutation that have been published in the literature. And then we've correlated the results of our experiment with the position of those mutations on the three-dimensional structure of the L36RA protein. And in doing so, we found some recurring theme. First of all, the mutation which map to the core of the proteins, which are buried inside, tend to affect the stability of the L36RA protein and therefore to be deleterious. And that, for instance, is the case for the serine-to-leucine substitution that is common among the European population, or the proline-to-leucine substitution that is common in North Africa. Then the variants that map to the interface between L36RA and the L36 receptor also tend to be deleterious because, of course, they disrupt the binding of the ligand to its receptor. Finally, the variants that map on the surface of the protein are a lot more likely to be benign. And we hope that this general principle will help us and others in the field to interpret new variants that are described in the L36RN gene as they emerge. So I've told you again that only 25% of GPP patients carry mutation in the L36RN genes. So there are other genes that contribute to the disease. And a couple of years ago, our group and the group led by Ulrike Hofmeier in Germany found that a small number of patients carry recessive alleles in MPO, which is the gene that encodes myeloperoxidase, a protein that is essential for neutrophil function. And you can see on the panel in the left, one individual who was homozygous for a C to A substitution. And then in the panel on the right, an individual who has a large deletion spanning 14 nucleotide and causing a premature protein truncation. And we know, we have demonstrated that these MPO mutations are associated with increased neutrophil counts in the circulation, which makes sense because, as you know, GPP patients tend to present with neutrophilia. And Ulrike Hofmeier's group also showed that these mutations result in increased activity of L36 cytokines. And although preliminary, these data are very interesting because they would reinforce the link between GPP and abnormal L36 signaling. So these mutations that I've just described to you, mapping to L36RN and MPO, are recessive allele, and we consider them as I mentioned at the beginning, sort of monogenic mutation. However, over the years, we've also found alleles that are not sufficient to cause disease, but can increase the risk of GPP. And for example, this is the case for these two variants that map to the AP1S3 gene and which are associated, we think, with roughly a twofold increase in GPP risk. And what is interesting is that defects in AP1S3 tend to upregulate L36 production. So you can see it here on the panel on the left. You can see that if we silence AP1S3 in vitro, this is the green bar in the graph, you see increased L36 production upon gene silencing. And then in the panel on the right, this is data obtained in patient cells where the production, so patients that carry this AP1S3 allele, the production of IL-36 cytokine is again higher compared to control. And so we think that AP1S3 alleles can increase your disease risk by increasing IL-36 production. And the role of this AP1S3 allele is also exemplified very nicely in this interesting case report, which is about two sisters we came across a few years ago. And one of them, you can see her on the top row, had GPP with very early age of onset. Her disease was relapsing and poorly controlled. The other sister, which you can see on the bottom row, had a single episode of GPP in her 30s and has since been well controlled with methotrexate. And interestingly, the sister with the more severe phenotype carry an IL-36RN mutation and an AP1S3 allele, whereas the sister with the milder phenotype only carried the IL-36RN mutation. So in this case, the AP1S3 allele was modifying the impact of IL-36RN mutation. And interestingly, in the literature, there are other cases of similar digenic inheritance. And we are beginning to think in the field that some individuals with GPP may really not carry any monogenic mutations at all, but actually more like a constellation of risk alleles. Another genes that we think increases the risk of GPP is called CARD14, and this is a very interesting gene in the, for skin inflammation because it has convincingly been associated with a number of inflammatory skin disease. In fact, CARD14 mutation have been observed in familial form of psoriasis, in pityriasis rubra pilaris, and in atopic dermatitis, to name but a few. And we found that among East Asian population, this particular allele, D176H, increases the risk of GPP. And interestingly, this allele maps in quite close proximity to other mutations that have been associated with other skin diseases. So, in conclusion, IL-36RN mutation are and remain the main genetic determinant of the disease. Alleles in other genes like AP1S3 and CARD14 can increase the risk of the disease or can modify the effect of IL-36RN mutation. And IL-36RN mutation are found globally in all ethnic groups, whereas AP1S3 allele we've only observed in European population, and the association with CARD14 has only been observed in East Asian population. Interestingly, IL-36RN mutation, AP1S3, and possibly MPO allele all upregulate IL-36 signaling. So there is a convergence of disease mechanism on the IL-36 signaling pathway. And this is why we think there is now overwhelming evidence that GPP is basically a disease of excessive IL-36 activation. Thank you very much for listening.